Hi everyone, I want to walk you through this week's virtual lab on tonicity. So I'm going to move down here to slide 10 where we see a graph of the lysis of normal red blood cells. So this whole experiment is centered around the structure of a red blood cell, which is normally sort of donut shaped. And as long as red blood cells are in a 0.9% salt solution, so this is a concentration of salt, then the cells maintain their shape. Once that concentration is decreased, say to 0.5 and 0.3, the cells change shape. With the lower salt concentration in solution, water is going to move into the cells and they begin to explode as they expand and explode. So that's called lysis, when cells explode. So what this graph is showing is at the very bottom here on the right, we have normal red blood cells. When the concentration of salt is high, it's not shown on the graph here, but high at 0.9 and 0.8, 0% of the cells have exploded. And as the concentration decreases, so as we move to the left, you can see that more and more cells begin to lyse. And by the time you reach 100% lysis, and you're right at the top, and the curve starts to plateau, you're at about a concentration of 0.3% salt. Okay, so on the next page, and on, here, on this slide, you are given a set of data. This is the same data that's in the assignment document. And so this is the absorbance of all of the different tubes that contain red blood cells in different concentrations of salt, okay? So we are going to measure their percent transmittance on the spectrophotometer, and the results are all written here. So the first thing you guys have to do, number one here, is determine the absorbance. So you're gonna you have a table in your assignment document. You can fill in the absorbance column by calculating 2 minus log of the percent T. Okay, so we can do the first one together. The absorbance at 0% concentration, so 2 minus log of 18.31. That's going to be 0.73. Okay, I have 0.7373 in my calculator. Oh, so I should round that up. You know what? Maybe let's take three significant figures. So we'll go 737. Okay, for my absorbance. So you can fill in the whole column in your assignment with the absorbances. The next thing we have to do is subtract the baseline. Okay, so the baseline is the absorbance reading at 0.9% salt concentration. So when you have your absorbance value for this data point, you're going to subtract that absorbance from every single one of your other absorbances, okay? So whatever this absorbance value is down here, you're going to subtract it from this absorbance, subtract from this absorbance, and so all of them are going to be, it's called normalized. We subtract the baseline from each of the absorbance values, okay? Then you can plot your graph. First you have to calculate your relative lysis. So you're going to divide all of your absorbance values by the highest absorbance that you have, and then multiply by 100. So you can fill in the column there for relative percent lysis. So all of the absorbance values that you have here in this column that have been normalized already, you're going to divide them all by one value, which is whatever your highest absorbance reading is, and then multiply by 100. So for all of this, all these values, you're using your normalized absorbances, the ones that you have subtracted. Okay, once you have your percent lysis, then you're going to make a graph with concentration on the horizontal axis. So this is your concentration of salt here, and the percent relative lysis on the vertical axis, okay, ranging from 0 to 100%. Once you have completed your graph, make sure you put in axis labels for your X and Y axis and put a nice title. And then you're going to determine the salt concentration for 50% lysis. So draw a horizontal line on your graph where 50% would be on your y-axis. Come over to your linear portion of your graph and then come straight down to the x-axis and estimate what that concentration is, that salt concentration. And that's it. You can submit your assignment in Blackboard as soon as you're done. Thank you.